share screen. Okay. Put that down here. All right, now we're ready to go. So just pretend like I didn't have to think that hard about that joke. That's pretty good though. Okay. The suspense was worth it. There was another one today too, but I had a lot going on this morning and I don't know what it was. I don't think I did. No, I did. I did tell you a joke. I did. All right. I have to bring some. I'll bring some next time because sometimes. Okay, here's another one for today. What letter of the alphabet has the most water? The C.
Oh. I thought you were going to say the Google letters for H2O.
No. Made you think that.
Keep us on our toes.
Yeah. All right. So we're skipping the end of chapter 6. It will not be on any exam. Very sad, but I think we got to move on, you know. And I don't want to rush through it and jam it down your throat. So I just think it's better to skip. So we are going to move on to chapter 7. Chapter 7 is all about enzymes, which is going to be really, really important because when we get to like glycolysis and the electron transport chain, and citrate cycle, and all that kind of stuff, you're going to have to understand how the enzymes work. And so this will give you that background. So 1926, this kind of always floors me, is when we discovered that enzymes were proteins, right? And we first crystallized them and actually looked at them and purified them. And so it took us a while to do that. And then initially we thought, right, that you learn this in biology, the lock and key thing. You've heard, how many times have you heard that? It's not true. It's not true. That's not how enzymes work. That was the initial explanation for how enzymes interact. But if you really think about it chemically, it doesn't make sense. And so we're going to talk today about why that is and exactly how the two are different. So the old lock and key tells us that the substrate fits the enzyme, right? You have this enzyme and it's this particular shape and my substrate has to fit that shape perfectly, right? That's a lock and key. It's not how it works. How it really works is what's called an induced fit. So this is our current model. Now there's nothing saying that as we learn more, we won't change our theories. But right now with all the data that we have, we can say it is an induced fit. So basically, remember we've talked about proteins being extremely flexible, right? They change shape all the time. And so what actually ends up happening is the enzyme is flexible and it changes conformation in order to accommodate the substrate. The substrate doesn't naturally fit in that pocket perfectly. So it will change conformations to accommodate that substrate. And when it does that, it's going to start making some weak interactions with the substrate. And so you can have an enzyme substrate complex, but the enzyme isn't designed for the substrate. The enzyme, here's the key takeaway. The enzyme is designed for the intermediate, right? Y'all know that in reactions, right? What you have is something that is a substrate, then you have a high energy intermediate, and then you make a product, right? This is not new news, correct? Okay, so what we're saying is that that enzyme actually has the most affinity for that intermediate. It doesn't have super high affinity for the substrate, and it doesn't have super high affinity for the product. Because if it had super high affinity for the substrate, it would never turn it into the product. If it had super high affinity for the product, it would never kick the product out, right? So it kind of actually makes sense. And we're going to talk about all the details on this and how it actually works today. So the first enzyme I'm going to introduce you to is hexakinase. So when you look at enzyme names, when you look at the name, it will tell you what it does. So what do you think hexakinase can do? It's a kinase, so it will what? It will phosphorylate. So the kinase, I always forget to press the ink button every time. So this will add a phosphate group. What is it adding a phosphate group to? A 6-carbon sugar, a hexose. What's an example of a hexose? Glucose. So it is going to phosphorylate glucose, right? So when you phosphorylate glucose, you actually make it charged and you trap it inside of the cell. So it's a really important step in making sure that metabolism will actually happen. That when the glucose enters the cell, it just doesn't get automatically transported back out. So it's really important. So if you look in A, this is the unbound state. This is the bound state. Okay. So in the unbound state, my molecule here is exposed to the solvent. What's my solvent in the cell? Water. Water is the solvent in the cell. So water can enter that enzymatic pocket. And right here is the enzymatic pocket. So when it binds glucose, it undergoes a massive conformational change. Now this is an exceptionally large change. Not all proteins have as large of a change, but it is a really good illustration of the concept that once glucose binds inside of that pocket, it induces a massive conformational change. Now we could even crystallize this protein, right? It's going to stay in this one conformation. So the actual binding of the substrate induces, that's why they call it induced fit, induces the conformational change. It's also going to exclude water from being in the enzymatic pocket. Because do you want this enzyme phosphorylating water? No. That would be a huge, massive waste of energy, massive waste of energy. So because glucose is here and it basically becomes buried, do you see how all of these side chains, all of these parts of the protein are basically surrounding glucose now? It's preventing solvent from actually getting into the enzymatic pocket. And so all the phosphorylation events are productive phosphorylation events, meaning that they will phosphorylate glucose. So where's the phosphoryl group come from? Comes from ATP. Comes from ATP. So whenever you see it in written in glycolysis and all that kind of stuff, what you're going to see is glucose is going to turn into glucose 6-phosphate. And what is that telling you? It went to the 6th, you put the phosphate group on the 6th carbon of glucose. And this is done in the presence of hexakinase. But I also have to show that this happens due to ATP, transfer of ATP, a phosphate group from ATP. So you start with ATP and it comes in and it comes back out as ATP. And so that's how you'll see it in the future. Yes?
At what point does the conformational change happen? When it binds to the intermediate or when it goes from the intermediate to the substrate? So the intermediate lasts for such a short period of time, but it's actually when it's making the interactions with the intermediate. And I'm actually going to show you that today. Yeah. Okay, so what this, if we use this basic theory, what we can say is that there are tons of different conformations that are possible for a protein, right? And we actually see that when we do things, when we look at structure in NMR, right? We get lots of different results from an NMR and we kind of put it together, right? But this is an illustration of that concept, right? Where it's the ligand binding is going to shift the preference for one particular conformation, right? And so then the molecule basically gets trapped by that ligand binding, and then we're going to increase the number of interactions that the enzyme is going to have. And it's those interactions that are then going to facilitate the actual catalysis from happening. What is catalysis? Catalysis is different from binding, right? Yeah. Catalysis is where we actually convert the substrate into product, right? So there's a difference between substrate binding and catalysis. And you have to kind of keep those things separate in your mind. So if we look at an enzymatic pocket, this is a different enzyme. This is phosphoglycerate kinase. So the substrate is 3-phosphoglycerate, and it's going to phosphorylate it. So it's not a big deal. But what we see here highlighted is the enzymatic pocket. And if you look in the enzymatic pocket is going to do a couple of different things that's going to promote binding and catalytic activity. So normally, if you don't have a substrate bound, what's in the pocket? Water, solvent. So the first thing that it's going to do in order to bind the substrate and promote catalytic reactions is to exclude solvent, right? So what we're trying to do here is we're trying to have this like perfect chemical environment for this catalysis to occur in, right? Do we want water in that perfect chemical environment? No, we just want an interaction between the enzyme and the substrate. So we have to get rid of everything else. So this is really the first step in the perfect chemical environment, right? Chemical environment. We're going to exclude the solvent. And the other thing that we want to do is we want to bring reactive functional groups together. Because you're going to have functional groups on the substrate and you're going to have functional groups on the enzyme. Where are the enzyme's functional groups? The enzyme. Where's the enzyme's functional groups? So all functional groups are going to be in the amino acid residues, whether we're talking about substrates or enzymes if it's a protein. But where on the enzyme are those residues? There's one portion of the enzyme where it binds, the active site. It's in the active site. So it's going to bring reactive functional groups. I go down of the enzyme. So this is in the binding pocket, or there's a couple of different terms for it, but I often call it the binding pocket. Binding pocket to the substrate, right? Because really the three-dimensional arrangement of the substrate in the enzymatic pocket is super insanely important. If that substrate is in the wrong position, are we going to have catalysis? No, right? And that's a basic chemical reaction thing, right? Things have to collide, and they have to collide in the right orientation. Okay, so when we have that substrate binding, so if we're unbound to our substrate, unbind, we have one conformation, bound, we have another conformation. So this is the same protein. This is hexakinase. So remember, hexakinase is the one that phosphorylates glucose. So if we have glucose unbound, we're going to have a particular conformation. And so this is the space filling. What you saw before were the ribbon diagrams. Now we have space filling. So what you can actually see are there are kind of two big arms to hexakinase. And they basically fold inward to protect the glucose, to keep it away from the solvent, and then to induce interaction of the functional side chain, which is really, really pretty cool. Looks like a what? A hug. It's hugging glucose. And then give it a phosphate group, right? Okay, so if we're going to have enzymes that can do these things inside of the cell, right, we're going to need to very, very, very highly regulate them. And there are a couple of different ways that we can regulate enzyme activity. One of them is just bioavailability. So how could the cell regulate bioavailability? There's more than one way. What do I mean by bioavailability? I mean the number of enzymes present that can do the job. How do we get proteins? Correct. So you can say transcription, translation, right? Right. So you could just say, or you could say AKA gene expression. Right. Those are the same two things. But it's how much protein are you going to make? So that's one thing that you can control. What's another way? Put it number 2 there. What's another way that we can control bioavailability? We can make it, and we can what? Not just to hide it. You could put it in particular compartments. You could sequester. Put them in something like a vesicle, something like that, right? You could do that. What's the other big one? Not just denature, but we're actually going to break it completely down into amino acids, right? So whether you're going to make it or you're going to break it down, either way is going to. So we call this protein turnover, right? How quickly is it broken down before it's made again? So this is protein breakdown, right? All those things control bioavailability. And that's completely separate from catalytic efficiency, right? Because if you break the enzyme down very, very quickly, even if you have a high catalytic efficiency, your enzyme's not going to be functional, right? Because you've broken it down, it's not there anymore. So you have to keep these two things separate in your mind, right? So let's talk about catalytic efficiency. I'll do that in blue. We'll do that in blue. So how can we control catalytic activity? What do we say was the on-off switch? Phosphorylation and dephosphorylation, but those aren't the only kinds. So I would just generally put covalent modifications. And one of them would be phosphorylation, dephosphorylation. And there are others, but those are phosphorylation, dephosphorylation are kind of the biggest ones. So that's one thing. So why would a covalent modification lead to a change in catalytic activity? So it changes the conformation of the protein. Anything that's going to change the conformation of the protein is going to change the activity of the protein. So the same is true about other regulatory molecules binding. So do you remember when we were talking about hemoglobin and we were talking about 2,3-BPG and we were talking about CO2 and O2 and all. So you can have binding of regulatory molecules. And they can bind close to the active site or they can bind far. So they can bind close or far from the active site. Because you can have an inhibitor that would bind in the active site and it would actually block the active site. You would reduce catalytic activity, right? But you could also have something that binds like on the other side of the molecule, but induces a conformational change that propagates to the active site that changes the conformation of the active site. Now that active site can't bind my substrate. So it could be close binding, it could be far away binding, but you can have all kinds of regulatory molecules that will bind. Okay. Kind of got lost on that one, right? Okay. So when we talk about enzymes, you have to think about catalysts, chemical catalysts. Enzymes are not the only catalysts. What are some other catalysts you learned in chemistry classes? Ribozymes can be, RNA can be, yeah. But like metals can be catalysts, right? Remember this, like, yeah. So you're always going to have something that will change the rate of your reaction, but will not change the ratio of substrates and products at equilibrium. What does that mean? If you have something that is A going to B, right? This is my general reaction that is going to be enzyme catalyzed. That means that the enzyme can increase A going to B. We can increase this rate. We can also increase the rate of B going to A. So you make the reactions go a whole lot faster, but you end up at the same equilibrium. That's really important to remember in enzymes. So you're changing the rate of the direction in both the forward and reverse, right? So rates affected, equilibrium unaffected. What is also unaffected? Delta G is unaffected. So the equilibrium rates, well, the ratio, the equilibrium ratios, and then delta G are completely unaffected, completely. So how do they do it? How do enzymes speed up either forward or reverse reaction? And it's all about the activation energy. So this is your typical energy diagram, right? How many times have you seen this in chemistry? Lots, right? Lots and lots. Okay, so this is my general reaction where I'm starting with my reactants. I need to overcome this initial energy input in order to transition to ground state products, right? So this transition state is the highest energy part of the reaction, requires the most amount of energy. So what do enzymes do? Am I going to change delta G of the reaction? No, so I have to start and I have to end at the same place, right? Because it's a state function, doesn't change. So what do I need to do? I need to lower my activation energy or change my transition state. So if I did this, this would be a catalyzed reaction. The red is my uncatalyzed reaction, right? So do you see this delta G double dagger? Do you see that? You probably know this as activation energy, EA, right? That's what most people, but biochemists do it a little bit differently. And so you'll see it as delta G double dagger. But if we do that same thing for our catalyzed reaction, look at the difference, right? So this is EA of our catalyzed reaction. How much smaller is it? Very. Much smaller. You have to go all the way up here, do that in red, all the way up here to get to our uncatalyzed reaction, whereas you only have to get up to this amount of energy for our catalyzed reaction, which is much easier to do because you don't have to input as much energy. And so this transition state, this high energy transition molecule lasts for milliseconds. And so you don't have very many molecules that are actually in that conformation at any given point in time. It's usually only a few. And then either you have reached the activation energy and you can go on and make products, or they revert back to reactants. But they don't hang out for very long because it's really hard to maintain them. And we're going to talk about them a lot. Okay, so most enzymes don't do their jobs all by themselves. They have helpers, right? And so they have either what are called cofactors or coenzymes in order to make the reactions happen. So cofactors are inorganic things. So they're small molecules that are usually within the active site. A lot of times they are ions, iron, copper, magnesium, zinc. You've heard of zinc finger nucleases, right? Why do they call them zinc finger nucleases? Because they require the zinc ion, right? So those are cofactors. They're inorganic. Coenzymes, on the other hand, are organic components. The organic components are a lot of times, especially in biochemistry, NAD and FAD. So if you are NAD, that's nicotinamide adenine dinucleotide, which means that you have two nucleotides. One of them is adenine and one of them is a flavin, which is a type of nucleotide that you don't really know. That's okay. And it's hooked together at the phosphate groups, okay? And then flavin adenine nucleotide is a flavin group and then an adenine nucleotide.
So is
Yeah, it is. It is. It is. But it's just,
I guess just like the generic version.
it's not the generic
Or
version,
name or whatever.
but it's the shortened name. It's the shortened name. So within coenzymes, you can have prosthetic groups. Prosthetic groups are actually covalently attached to our enzymes, and they can't ever leave. Coenzymes can. If you don't have enough NAD+, you don't have enough FAD, your reaction's not going to go. But if you have a prosthetic group, it is hooked on, it's attached, it's never leaving. So you can have different things. So whether you have a cofactor or a coenzyme, whatever it is, if it has everything it needs, so if it's with cofactor, coenzyme, it's active, and we call this active enzyme the holoenzyme. Sounds like whole enzyme, but it's actually holoenzyme. But that's how you remember. In order for it to run, it needs everything. It has to be whole. So it's the holoenzyme. If it does not, if it's without whatever it needs, therefore it is inactive, we call that the apoenzyme. Okay. So holoenzyme and apoenzyme are the two. All right, so when we look at some of these, when you look at the names, right? A lot of times what you can do is you can look at the name and you can predict what its role is going to be. Do you have to know what the cofactors are for representative enzymes? No, you do not. Do not go forth and memorize that. Please do not. But what I want you to be able to do is to look at an enzyme name, cytochrome oxidase. If it's an oxidase, it's going to participate in an oxidation reduction reaction, right? Hexakinase. Kinase is going to facilitate phosphorylation, right? So this cofactor here is actually going to help ATP to bind in order to facilitate that phosphorylation event, because you actually have to recruit ATP in order to do it. So just being able to look at them and knowing generally what they're going to be doing just by looking at the names. Lots of oxidases, dehydrogenases, all those kinds of things. So let's look more specifically at NAD+ and NADH. So this is a cofactor. If you, sorry, no, they just show cofactors here. They don't show coenzymes. So this is a coenzyme, right? Because this is organic. So this is our coenzyme that is going to be important for many different reactions within energy transfer reactions that we're going to talk about like so much. So here's our example, right? So NAD, nicotinamide, adenine dinucleotide means we have two nucleotides. This one is adenine, right? And this one is nicotinamide. And then they're hooked together by phosphate groups, right? But you don't have to be able to draw it. I just want you to know where the name comes from. So what this can do is this can actually pick up electrons, right? And that is all about oxidation reduction reactions. All they do is they pass along electrons. And we're going to see that a lot in the electron transport chain. And so what you can see is the NAD+ is actually going to pick up on the nitrogen right there, those two electrons. So we go from the oxidized state to the reduced state. So you remember your oxidation reduction rules? What's your little mnemonic? Do y'all do that here? Oil rig. Down here, we better do oil rig, right? Oxidation is loss, reduction is gain of what? Electrons. So what's happening as we go from here to here? Is this an oxidation reaction or a reduction reaction? It's a reduction reaction. Reduction reaction. And so we're going to see this a lot of times, but if you don't have an electron carrier, even if you had the enzyme, the process isn't going to happen. So you need this cofactor in order for all of these reactions to occur. Here's an example of our coenzyme, but this is a coenzyme that is actually linked to our molecule, right? So this is lipoamide. So here we have a lipo group that's actually connected to, here's our polypeptide chain. What functional group, what side chain are we looking at here? Lysine, right? But it's lysine that's actually been bound, so now they call it a lysyl group, right? But we can convert from being oxidized to being reduced because this lipo group has two sulfurs, right? So just like in a disulfide bond, the disulfide bond can be oxidized where you have the bond between the two sulfurs, or it can be reduced where we have now picked up two hydrogens, and so you're unbonded. So we're going to see that as another example of a coenzyme, right? And you'll see this a lot in processes that require a decarboxylation reaction. You'll see this. And we'll show some examples when we get to it. But here's our generic enzyme nomenclature. Most enzymes are going to end in ASE. If you see it end in ASE, it's an enzyme. And then usually we include the substrate name. And a lot of times we say what class of enzyme it is in the name, right? So hexakinase, we have the substrate, we have, what is it doing? It's a kinase, so it's adding a phosphoryl group, and then it's an ASE, so we know it's an enzyme. So if you look at the name, you should be able to predict what's going on. So what I would actually recommend is actually really studying this table. Because when you look at the names, you're going to want to be able to recognize what's happening, right? So remember we said carboxylases, right? The enzyme class is a lyase. Why do we call it a lyase?
The lysyl group?
It's attached to the lysyl group, right? And we have the lipoamide group there facilitating that. So just kind of look at them, right? A ligase, if you're going to ligate DNA, what are you doing in DNA? We talked about this when we were making plasmids. Restriction nucleases cut it, ligases seal it back together, right? So it's a similar thing. You're actually going to form a new bond instead of break a bond, right? And they usually do it with ATP, so they call those synthases. But isomerases, right? Isomerases, you're gonna whatever molecule you started out with, if you look at the molecular formula in an isomerase, you're gonna have exactly the same molecular formula, but you have a different substance because all you did was rearrange the atoms, right? So it's intermolecular rearrangements, things like that. So go in, study those. It'll be really, really helpful. Well, it will just be helpful in the long run for you. It really will be. So why do enzymes facilitate substrate to reaction changes? And so they're gonna have three different impacts on chemical reactions. Number one, we said they lower the activation energy. But how is it that they are actually lowering the activation energy? Usually these intermediates are super unstable, right? So usually there are parts within the enzyme that act to stabilize those unstable parts of our intermediate, right? So if you have lots of negative charges on your intermediate, what do you think the enzyme is going to have? Positive charges to balance them out so they're not so unstable. They can also provide an alternative path for product formation. So you can go through a different set of chemical steps in order to get to the same product that would require less energy. And then they actually reduce the entropy because if you have a positive part of your enzyme and a negative part of your substrate, the substrate is going to go into the enzymatic pocket in a particular orientation. And so it's going to orient those substrate molecules so there's less movement of those substrates, lock it in, and now I'm going to bring in the other substrate and cause that chemical reaction to occur, right? So it helps them to orient in the right way so you have less entropy. for that. So if we look, this is just a pictorial of exactly what we said, right? In this, we're going to start with two substrates, substrate A, substrate B, and we have our enzyme. So if you look, do you notice how we have different parts here? So we have pluses and we have minuses. What does that refer to? That's just portions of the substrate molecule that are charged. And then inside the enzymatic pocket, we also have parts of the enzymatic pocket that are positive and negative. We also have hydrophobic and hydrophilic regions that can align in the same way. And so that's exactly what happens is these substrates come in and they orient themselves in a precise way so that, okay, everything that's necessary for that active site, right here is the active site. This is where the chemical reaction is actually going to occur. That all of this stuff lines up perfectly so the two molecules or the two components are in the right orientation and you can have that chemical reaction proceed very easily. So you're going to have charged residues within the active site a lot of times, and you'll also have those charged or hydrophobic or things like that, those weak interactions that are going to occur away from the actual active site. Okay. And then the enzyme induces a conformational change that facilitates the change in the substrates and we get products, right? So we have a couple of different things if we look at this that are actually happening, right? If you have these two molecules that are coming in close contact with one another, basically what you're doing is you are increasing the concentration of those two things in that very, very immediate local area, right? And you remember in like General Chem 1 or 2, right? What are the things that speed up a chemical reaction? One of the things was concentration of substrates. So in effect, what you are doing here is you are increasing your local concentration of substrates. Just because you've put them in the right orientation next to each other using the enzyme, you've increased their local concentrations. You have portions of the molecule that have high affinity for one another, right? So our positives to our negatives to our positives, are hydrophobic to hydrophobic, right? So you have local regions of enzyme substrate that have high affinity for one another. High affinity for each other. So why is that important? Why do we want local regions that have high affinity for one another? What does that give me? Alignment of what? Alignment of the actual functional groups that are going to participate in the chemical reaction, right? Because if we get the substrates in the correct orientation, now those functional groups that are going to actually participate in the chemical reaction is not going to be the whole molecule. It's just a little piece of the molecule. And so by orienting them in the right place, we're going to alignment of functional groups that participate in the reaction. Right? Because the functional groups that are participating in the reaction are right here. Are they these pluses and minuses? No, those are just for alignment. All of this is just for alignment. The blue circle, that's where you're actually having enzymatic reactions occurring. And then the last thing, right? So physical and chemical properties of our active site, we're going to increase our local concentration. We're going to align our functional groups to participate. And then the third thing is we actually have to have functional groups that will participate. So this is like 1, 2, and then 3 is that we have functional groups that participate in the chemical reaction. I know, it happens. It happens because I'm left-handed. That's what happens. It's hard for me when it gets to that end of the screen. This end is easy. This side's more difficult. I feel like it's better for me if I write it down for y'all, right? Yes. I feel like I talk a mile a minute. So sometimes it helps slow me down a little bit. All right. So if you don't have those three things, right, you're not going to be catalyzing anything. All right. So this is kind of what we were talking about, right? Sequesters are microenvironment of our active site. So this is exactly what we were saying earlier, in that we have this perfect alignment of substrates because of how the molecules are fitting. And once they're aligned, is there room for the solvent? No, right? So you're in effect increasing your local concentration of your substrates and excluding everything else, which is awesome. And then you have interactions between the substrate and the enzyme. And I'm going to show you some examples, and it's going to make way more sense. But you actually have interactions between the side chain residues of the enzyme and the substrate itself. And you make a transition state molecule. You turn that substrate into a transition state molecule. And then you have those functional groups that are participating in the actual catalytic activity. So here's a real picture of what happens when you have a substrate binding to an enzyme. You don't really just have this decrease in the hump, right? What you actually have are a few smaller humps that are occurring. So let's take this and let's kind of tease out what's actually happening. So what happens in our very first step? We have enzyme plus substrate, right? The enzyme is going to bind to the substrate. So how would I write that if this were a chemical reaction? And remember, almost everything is reversible. We're going to make enzyme substrate right here, right? Right there. Whoa. Okay. So once we have the substrate bound to the enzyme, now we're actually going to convert that substrate into product, right? So that's really another reaction where we're going to convert that into our enzyme product, right? But are we done? No, because we don't want the product still attached to the enzyme. So now what's our next chemical reaction? We need to separate it into E plus P so that it will kick out the product and be done and be ready to go again. That's the other thing that's really important about enzymes, right? That by the time they're done with their process, they're back to their original state and can bind another substrate. So you have to be very careful about that. So let's talk, this is actually 3 reactions. Do you see how this is 1, 2, 3 reactions that are actually occurring? Yeah. Yes, but it's very, very, very small. And we're actually going to talk about that in the next section. It's actually extremely small. And so a lot of times we just assume it's a forward. But there are cases where it can go in reverse. So we like to draw it like this if you're not specific about the reaction. But we will assume in the future that it is primarily forward. Okay, so in order to make this reaction happen, reaction 1, we're going to have some kind of interaction between the enzyme and the substrate, right? So we're going to have many weak interactions. I'll say BT is my abbreviation for between enzyme and substrate, right? So this could be electrostatic interactions, positive and negative. This could be the hydrophobic effect, right? Weak interactions. What happens next when we go from enzyme substrate to enzyme product? Oh, this is not gonna be easy. Because in order to convert from the substrate to the product, we have to be making and breaking bonds, right? So this is the step where we actually make and break bonds. So what actually ends up happening is that the substrate will converts to the transition state. whoa, converts to transition state. If I could really spell, that would be great. So you have lots of interactions between transition state and enzyme. And this is super important that you have so many interactions with that transition state, because what that's going to do is that's going to lower the amount of energy that you have to input into the system to make the transition work, right? Normally this transition molecule is hugely unstable and requires lots and lots and lots of energy. Move it down. But because we have an increase in the number of interactions between the enzyme and the transition state versus the enzyme and the substrate, you're actually lowering the amount of energy that has to be put into the system to make and break those bonds. Can you tell I think this is really cool? Okay, so our last step, right? Our last step is going from our enzyme product to our enzyme plus product. So the product here is released. That's what we want. Oh, this pen is RELE, released. Okay. And why is the product released? Where did we say we have the highest amount of interaction? Between the enzyme and, is it enzyme and substrate, enzyme and transition molecule, or enzyme and product? Transition molecule. It's absolutely our transition molecule. So because it's higher affinity towards that transition molecule, once that transition molecule converts into a product, the enzyme's like, see you later, there's something better coming along, right? So it actually kicks out the product because it has higher affinity for transition molecule, right? Because we said they don't stay for very long. So you'll have another transition molecule come in, right? It's like a chain. When you kick out the product, something else is going to come in, and it's going to start all over again. So that's what this really means. Yeah? Questions on this? Go back. Did you copy everything? Yeah? Okay. All right. So how do we know this? Right? I think that's a very valid question. How can we be so sure that the molecule has such a high affinity for the transition state versus the substrate? Well, what we can do is we can create what are called transition state analogs. So there are molecules that look a whole lot like the transition state, except that they're stable. So for example, if we have this, we have adenosine deaminase. An adenosine deaminase will deaminate adenosine, right? So our proposed transition state conformation, we can't really check because they last for such a short amount of time. It is really, really difficult to know exactly what their shape is, but this is the proposed transition state conformation. And so what we're really looking at, right, is right here, we're converting this group from an NH2 to a double bond to an oxygen, right? So this is basically where on the adenosine, we're gonna have the chemical modification. So in the transition state, this carbon right here, can you see that it's a tetrahedral shape? And we have a hydroxyl attached to it. You have to have those things in the transition state because we're trying to kick out the NH2 and keep the oxygen, right? So we create this analog. Here's our analog. What do you notice about the analog? Instead of an NH2, it's an H, but we still have a tetrahedral and we still have the hydroxyl group. And these are the portions of the molecule and the conformations of the molecule that are important for substrate activity. So we have that same tetrahedral and we have the same hydroxyl. So the whole molecule isn't the same, but the important bits are. And so if we look at just binding affinity, for the enzyme and substrate, what you're going to see for the normal substrate is something like 10 to the negative 5, 1 times 10 to the negative 5. That's pretty good. That's pretty high affinity. Binding affinity for the enzyme plus the analog, 1 times 10 to the negative 13. That's a huge difference. That is an insanely large increase in binding affinity. Insanely large. So we can actually, this is the really cool part, you can use medicinal chemistry to actually treat biochemical disorders, right? Because I can block an enzyme with this. If you have higher affinity for this analog and it's stable, it'll hang out in the body, that's great. It's going to block this enzyme that I want to block for whatever reason, right? So I can use this as a drug design tool. Isn't this awesome?
So the 10 to the negative 5 and the 10 to the negative 13 is the difference in activation energy?
In binding affinity.
So the lower the number,
The tighter the binding, because remember, this is your KD. This is your dissociation constant. So are you going to make very many disassociated, right? You go from enzyme plus substrate to enzyme substrate. That's the reaction we're talking about. But really, what are you measuring? You're measuring KD. KD is of enzyme substrate going to enzyme plus substrate, right? So that's KD. So if you have a very small K disassociating, then that means a very high affinity.
okay, so the lower the number, the higher.
The lower the number, the higher the affinity, right? Yes.
You said that the enzyme stabilizes the mid-transition state. How does that read into this? Like, does that decrease affinity?
I'm going to show you.
Oh, sorry.
No, it's good. That's an awesome question. It's an awesome question. So I'm actually going to show you a couple of different mechanisms. So in order to understand how the enzyme interacts with not only the substrate, but also the transition molecule, you have to look at the catalytic functional groups, right? Because that's what's actually going to be participating in the interactions. And so remember our amino acids. You remember how I made you memorize the amino acids, how terrible and how mean that was of me. But it's really, really important because what you're going to see is that there are really 3 different ways that the active site will actually contribute to catalytic activity. So we're going to say 3 ways active site contributes to catalytic activity, right? So you're going to get the substrate into the enzymatic pocket. And so the amino acids on the enzyme are actually going to interact with the substrate, and it's going to catalyze the reaction. So there are 3 different ways that catalysis can actually happen. And so the first one is called acid-base catalysis. So if you look at the enzymes on this slide, what do you see? An acid form and a base form. Oh, okay, so that's one of them. I'm going to tell you the other ones, but we're going to go over all of them eventually. And if it's acid base, there are actually 2 different types of acid base. There's general acid base, and general is for all amino acids, right? There are lots of different amino acids. So if I say general acid base catalysis, I'm not telling you which amino acid. I'm just saying that amino acids make it happen. The other one is specific, which means it's always going to be water, right? There's only one possibility, it's just water. Okay, so you could catalyze through acid-base interactions. You could catalyze through covalent interactions. And then the last one is through metal ions. And so what we're going to do is we're going to go through and we're going to talk about each of these individually. And I think from the packing up, it's time. So we'll do that next time. Y'all have a good rest of your day. I'm working on grading exams. I will get them done as soon as I can. I'm working on it. Okay, anybody on Zoom need anything? Good. All right.
They stop research.